good evening, everyone. Welcome to the Alumni lecture. We are currently figuring out the screen and the camera, so I hope that me and Mike, of course, will be visible. Tonight we're going to listen to Professor Mike Jette. He is a world-leading expert in environmental microbiology, and he has been chair of our very own Department of Microbiology here at the Radboud University since the year 3000. Besides 2000. Year 2000 is a song, very good song. Tonight, I first want to further introduce you. Besides having multiple ERC advanced grants to its name, Mike Jette has been invited over 250 times for keynote lectures, and he has organized his fair share of international conferences and meetings. So we are very honored to have him here tonight to host the last alumni lecture of the season. Tonight, Mike is going to explore how microorganisms can actively form a methane biofilter and could possibly help to counteract climate change. So Mike, I'm hoping for a hopeful message tonight. I would love it if I don't have to move to Germany in the next 30 years. So my hope is invested on you. So if you have any questions during the lecture, feel free to ask. You can also save them for the end. Also for the people in the live stream, I will figure out how to see your chat and then make sure that Mike gets your questions. Just want to make sure that everyone knows that we need to stop at 9. That's also when the live stream will end. So it would be great if we can wrap everything up before 9 so the people in the live stream don't get cut off from any information. So if there's any questions left after 9, please just save them for when we're having drinks and you can ask Mike yourself. So without further ado, Mike Jette.
Thank you, Merel, for the introduction, and thank you, Alumni, for the invitation. It's an honor and pleasure to bring you up to date on microbial methane and nitrogen cycle. And then maybe Merel didn't mention the award that I'm most proud of is Supervisor of the Year, because that was a surprise and was made by the PhD candidates that I supervised. What do we have in stock this evening? First, a crash course, microbiology. I see a few people who don't need it, but maybe most of you, it was a long time ago. Very welcome. Just in time, we are about to start a crash course in microbiology. 7 to 8 minutes, then I'll present one example in the nitrogen cycle, a few examples in the methane cycle, and then we wrap up with some conclusions and outlook. That okay with you? First and foremost, I would like to thank our past and present collaborators, co-workers, collaborators, and funding agencies. Dag meneer de Dekker. And you've seen since 2000 we have quite expanded in size. A lab day out in 2000 and a lab day out 2 weeks ago, I think. Yeah. Oh. Nitrogen. All of you have been following the news, know that in the Netherlands we have both a chronic and an acute nitrogen crisis, cycle crisis. I've depicted that here in the cartoon. This is what we do in the Netherlands. We fertilize our agricultural fields with 200 kilograms of nitrogen per year. Due to microbial activity, a lot of this nitrogen is lost as nitrate into the groundwater, and then about 20% is captured in crops. For husbandry, it is even worse. We spend 450 kilograms of nitrogen on raising the animals. A lot of it comes out as shit, manure. then microbial diffusive emissions, ammonium nitrate, and about 14% ends up in meat. Especially in these diffusive processes, microbes play a great role, and the leaching of nitrite leads to eutrophication of many ecosystems and loss of biodiversity. Maybe what is less known, all the conversion of ammonium requires a lot of oxygen, we will see that also later, leading to deoxygenation, loss of oxygen in ecosystems, and especially freshwater marine systems. Fish do like oxygen, and they don't like ammonia. During these two emissions, nitrous oxide, laughing gas, is emitted, the third most abundant contributor in climate warming. Luckily for us, then the good message for Merel, we can also use microbes to remove nitrogen from wastewater and that it is cleaned before it is returned to surface waters. For methane, also in the news, but maybe because of its price as a biofuel, going skyrocket today. In addition, methane is a greenhouse gas. 16 to 25% of the global warming is done by methane. And that's also, and since methane is not so very long-lived in the environment, if we cut methane emissions, we will have effects within 10 years. So slashing methane, cutting methane emissions is a target that we should strive for. As a microbiologist, I have maybe a peculiar view on the world. We live in a microbial planet. And these microbes are also on this planet for a very long time, already for nearly 4 billion years. And during the first 2 billion years of our evolution, there was no or hardly any oxygen available for the development of life. So all developments, the first 2 billion years, were done without oxygen. And still today, there are many important ecosystems without oxygen. Already I see 20 ecosystems here in the room. Our guts are nearly completely devoid of oxygen, a very anaerobic ecosystem. Oxygen minimum zones in the ocean, anaerobic sediments, anaerobic wastewater treatment plants, they are all important anaerobic ecosystems. And all of these ecosystems are characterized by the same anaerobic food chain. If we start on the top, we have the polymers, polysaccharides, proteins, lipids. They are degraded to their respective monomers, sugars, amino acids, fatty acids, and then they are fermented to organic acids, acetate, propionate, a very important compound. There it is again, ammonium, hydrogen, and ultimately all the carbon ends up in methane. As microbiologists, we have still a lot to discover. We estimate with molecular techniques that at the moment we know about 2 to 5% of the microbial diversity on our planet. And that means that we still have a large piece of the pie to discover. And that was maybe also the basis why in 2014 we got a gravitation grant to discover these new anaerobes. So we were able to found the Zeuntgen Institute of Anaerobic Microbiology with a gravitation funding. And there we want to discover new anaerobes that can improve our health. That's mostly done in Wageningen, new anaerobic. And I see in September they will launch their first product, Akkermansia, a new bacterium against obesitas. Microbes to slash the methane, and we want to be the basis for a bio-based economy. No. Oh. It's either working too slow. No, come up. All right. Oh, I said 95% of the diversity we still have to discover. You cannot discover them all, so we had to make some choices. If you look at the diversity in metabolism, Several microorganisms, cyanobacteria, algae, can harvest energy from the sun. They studied that in Amsterdam, not here, so that's another expertise. Many organisms can use organic molecules. Wageningen and Groningen are more specialists in that. And here in Nijmegen, Radboud University, we study the conversion of small inorganic compounds by microorganisms. So ammonium, methane, hydrogen, chemolithoautotrophs, as the Greek name says. And when in 2000 we moved from Delft to Radboud University with our team, we made a kind of periodic table of missing chemolithoautotrophs. And here is this periodic table. In the top row, you see the electron donors, the energy-rich compound that drive the reactions, hydrogen, methane, hydrogen sulfide, ammonium, and iron. And if you would look into the top layer of a sediment, freshwater sediment, there would be oxygen available, and hydrogen and oxygen, a very potent mixture, canal gas. The organisms can live from this canal gas. There is enough energy in this reaction that they can conserve energy and grow. Now if you go deeper into the sediment, when oxygen is depleted, they could use nitrate as an alternative electron acceptor, metal oxides, sulfate, and finally when all the electron acceptors are gone, they ferment and could make methane. The green boxes indicate that we more or less know which important microbes catalyze these reactions, but in this periodic table there were two groups of organisms that we really wanted to get our hands on. If start with methane, if you can burn methane with oxygen, then also in nitrate, there is more than sufficient Gibbs-free energy to catalyze this reaction. So we were really wanting to know which organisms can burn methane without oxygen, so anaerobic methane oxidation with nitrate. And the other one, wouldn't there be organisms that could oxidize ammonium without oxygen, but with nitrate or nitrite? Of course, easier said than done. People had been looking for these organisms for 50 years. Many trials, more errors, and then they got the name in the literature, impossible microbes. Maybe that's more a qualification for us as microbiologists. Surely, for every negative Gibbs-free energy, a microorganism exists that can harvest that energy. But maybe we have to learn better how to get them. And a very wise man said, it always seems impossible until it is done. And we took up the challenge to find these organisms. Especially when there is a camera, we go out in the field. Or I go out in the field. My students do that also when it rains. We take samples and we try to enrich the organisms. We do that in bioreactors under very controlled conditions, trying to mimic the ecosystem as good as possible, meaning as little oxygen as possible, sufficient ammonium or sufficient methane, and very slowly dosing of the substrates. That's the key, very slow, always under starvation. That's what they see in nature too. In nature, organisms don't have enough food, so normally we feed way too much, and here we try to give as little as possible feeding. Once we have an enrichment culture, we can nowadays unleash the genomic toolbox, sequence their genomes, and see what metabolic secrets they would have. Then we can do biochemistry, advanced microscopy, and with this knowledge we can go back to the ecosystem and see if the organisms are really important and if they are doing the job we think they would be doing. No, nothing works anymore. Go back to the anaerobic food chain. The polymers end up in ammonium and methane. And what is the fate of this ammonium in the ecosystems? Normally it's used for microbial biomass, that's fine, it's removed from the ecosystem, microbes can go. If it leaches into the oxic layers, it is oxidized with a lot of oxygen into nitrite and nitrate. Both ammonium and nitrite are toxic to aquatic life. It leads to the consumption of oxygen, hypoxia in ecosystems. During these processes, nitrous oxide, laughing gas, can be emitted, contributing to climate warming. And thus it would be very beneficial if we could find organisms that can combine the ammonium with the nitrite and make dinitrogen gas. Luckily, it was already Engelbert Broda. He is a theoretical biologist in Vienna. He wrote a very nice paper in 1977 already predicting that this organism must exist. Engelbert Broda had, as a there are probably theoreticians in the room, but he had too many time on his hands, and in the evenings he spied on his nuclear physicist colleagues and sold it to the KGB. You can follow up this link and you will read his biography. About 20 years later, Arnold Mulder was operating an anaerobic wastewater treatment plant at that time Gist Brocades, nowadays DSM, a yeast and penicillin factory in Delft. During the process, a lot of sulfate-rich wastewater was created, and when you treat sulfate-rich wastewater, you produce a lot of... Sander, jij weet het nog wel. Under anaerobic conditions, what happens to sulfate? Yes, very good. You make hydrogen sulfide. Hydrogen sulfide, very smelly. Yeah, very smelly. So the citizens of Delft complained to Arnold, Arnold, please change your wastewater treatment system. Arnold was a very pragmatic engineer. He bought copious amounts of calcium nitrate, added the calcium nitrate to suppress sulfate reduction, and inadvertently he had created good conditions for anaerobic ammonium oxidation. And you can also see about half a year after the addition of calcium nitrate, he saw that ammonium in the effluent was disappearing. There was less ammonium in the effluents. And luckily, Arnold had read the paper of Engelbert Broda, so you know, he connected that there would be anaerobic organisms removing ammonium. He came then to the TU Delft, to Kees Kuhnen, Astrid van der Graaf, and I joined them at that moment, 1992. And together we went on about to discover which organisms were responsible for this ammonium removal. And I'll make a very long story short in a few slides. What do you do if you want to study a process? You take your students to a very inspirational place. For us it was the Oktoberfest in Munich, 1995. And you really get good ideas there, especially on the way back in the train. We came up with a bioreactor system in which we could cultivate the Anamox bacteria, a sequencing batch reactor, which is very efficient in keeping the biomass in the reactor and keeping the concentrations of the nutrients low. And maybe the Dean knows, and some of you will know, we have published in glamorous journals many times. But if you do a very good discovery, you don't need to go to Nature or Science, and you can get a paper in a journal of your field, and this is the highest cited paper we have. So that is something very useful for the field to develop a reactor system. Gave us a lot of biomass to do experiments, but nowadays we prefer to have a membrane system where the cells grow suspended without any polymers, and they grow quite slowly for bacteria, doubling time of 10 days. In the membrane reactor we have cranked it up to 3 days. But remember, it's an anaerobic bacterium. It does not like oxygen. Ultimately, we were able to purify the Anamox cells to a homogeneous suspension. that was published in a glamorous journal. And once you have a pure suspension of bacteria, you can extract their genomic material, their DNA, and on the basis of DNA you can make diagnostic tools, PCR primers, certain probes to fish for these organisms in the environment. So once you know the genetic material, you can go out in the world, and I said anammox is everywhere, where there is very limited oxygen and a lot of ammonium, you will find these organisms. And I see two biology students in the room. Your predecessors, they had the opportunity to do an internship in Uganda, sampling several African lakes, and there, when they took the samples back to Nijmegen, they made one of the most beautiful Anamox pictures, microcolonies of Uganda Anamox. Unfortunately, we could never enrich them in our lab, but we have a very nice picture. Oh, and now it does again. All right, to make a long story short, we wanted to know how Anamox works. A lot of trial and error, and this is the end result. If we start with nitrite, Anamox reduces nitrite in a 1 electron reduction to the level of nitric oxide. Then we have a very intriguing complex of hydrazine synthase, which combines nitric oxide with ammonium in a 3 electron reduction. The spectacular intermediate hydrazine rocket fuel is made, and then the final step, this rocket fuel is oxidized to dinitrogen gas, releasing the 4 electrons at a very low electron potential. They go into the electron transport channel the electrons back to the other reactions, make sure that the protons are pumped across the membrane, and the protons are used for ATP synthesis. Especially the intermediate hydrazine has inspired journalists to write about the Anamox process. I'm not sure if you have to believe everything what is written in the newspaper. I'm not sure that astronauts can pee enough and make enough urine to be converted to hydrazine to get a rocket to the moon or even Mars. But who knows in the future? We were intrigued. We wanted to know which enzyme complex would be responsible for the production of hydrazine. So we had sequenced the genome of an Anamox bacterium, and then Anamox is mostly a difficult organism, but for once it was not too difficult. We looked for an enzyme or a gene system that was not known in other bacteria and highly expressed, and we could find a gene cluster of 3 genes that was really expressed through the roof, and this turned out to be the hydrazine synthase genes. If you are highly expressed messenger RNA, you probably also make a lot of protein. About 10 to 15% of Anamox protein is the hydrazine synthase. And together with the group of Thomas Beirens in Heidelberg, we could crystallize this protein. We now know the crystal structure and have an idea about its mechanism, but we are still investigating this together with Thomas to find the molecular details. From the hydrazine and the fundamental science to the more applied science, if we look at wastewater treatment in the Western world, in the morning we all go to the toilet. All our excretions, urine, feces go into the sewer system, become very, very diluted, transported in our case to Wörth. There the ammonium and the organics have to be removed again, and this process, especially in diluted, remove something from a diluted stream is a costly process. Therefore we need a lot of oxygen to oxidize the ammonium into nitrate. Then we reduce the nitrate either with the organics in the sewer system, or most of the times you have to buy methanol to remove the nitrate, and unfortunately you make a lot of CO2 and nitrous oxide during that process. Now, if we could implement the Anamox process in a wastewater treatment system, what we could do is we could save 75% on oxygen, because we use little oxygen to make a mixture of ammonium and nitrite, and this is then converted to dinitrogen gas. We would also prevent the use of methanol and CO2 production. Anamox is actually a CO2 consumer. no nitrous oxide, and much less biomass. And in about 2002, we set up a collaboration with the Park Biotech company in Balk. Balk took a license on the process, and they built the first Anamox plant in Rotterdam. And later on, they exported it to China. Biggest plant is in China. Recently, they built a Rendak plant in Sonnenbreugel for treating meat processing waste. It's the biggest plant in Europe. And also, unfortunately, the competitor of Park built a Anamox system in Weert 2 years ago. And by implementing the Anamox process, the energy balance of the Nijmegen plant has become from an energy-consuming wastewater treatment system into an electricity-producing system. So they have an excess of electricity. Not too bad. The take-home message from the nitrogen cycle is that it is an important cycle with many microbes participating in it. We have found the Anamox bacteria, and they use the rocket fuel hydrazine as intermediate, and Anamox is already implemented in wastewater treatment system, making it much more sustainable. But of course, we don't know yet everything about Anamox, and luckily, the current chair of the Department of Microbiology, Laura van Ifterik, first of all, she got from Cyber and the faculty, together with Daniela Wilson, a seed money to look into this hydrazine. And further, she was also chosen to be in the Radboud Fonds. The Radboud Fonds is eagerly awaiting your contributions. If you scan the QR code, you can donate the money to Laura's research. Highly recommended. Oh. What do I foresee in the future in the nitrogen cycle, not only in wastewater treatment systems? Yes, you can convert ammonium into dinitrogen gas, but we have enough dinitrogen gas in the atmosphere. That's maybe not the most sustainable thing to do. People are trying to develop a phototrophic process in which phototrophs capture the ammonium, and that maybe could be used as single-cell protein. People are also trying to precipitate the ammonium as ammonium sulfate or struvite, where you could then feed that into processes. Ammonium as an energy carrier is at the moment developed, especially for very big ships. Ammonium has 30% more energy per volume than hydrogen gas, so it would be, especially for these ships, a good energy source. And already there are oxidation processes with as little as possible NOx emissions. And one of the things we are ourselves working on is that Anamox bacteria can also convert ammonium into electricity. So if you have a concentrated seam, then Anamox bacteria could, on a local scale, convert the ammonium into electrons, and that could make local electricity. I don't think on a very large scale, but that's something we are investigating. Enough about nitrogen. We'll move on to methane. Very scary curve, methane, from pre-industrial times to now. Already this year we exceed 1900 ppb in the atmosphere, contributing 16 to 25% to global warming. And as I said, because methane is not long-lived in the atmosphere, what you reduce now has an effect within 10 years. So that's why also 30 countries have pledged to reduce their methane emissions by 30%. The more the better, but it's at least the beginning. Cornelia Welt is the group leader on the methane research here at Radboud University. We tackle a number of processes. There are several anaerobic microbes that can produce methane. And in fact, I think 80 to 85% of the methane that we use is of microbial origin, made long time ago from dead plants. We study methane emissions from wetlands, rice paddy, urban waterways, coastal systems, and also from permafrost, a time bomb. Permafrost is full of methane. If it warms up, the methane is released. Methane contributes to global warming. and it's a positive feedback loop giving more methane. And hopefully the microbes, Merel, your message, hopefully the microbes can help us to remove that methane before it reaches the atmosphere. Here again, this periodic table, now this segment for methane. It was already discovered in 1906 by Nicolaas Söntgen that methane could be oxidized at the expense of oxygen. He discovered Bacillus mechanicus in the canals of Delft. And since 2000, we knew that also with sulfate, methane could be oxidized anaerobically at the bottom of the sea. So if both oxygen and sulfate are possible, then certainly nitrate would be a good source. Now, if you want to find nitrate-dependent methane oxidizers, then you should go to places where there is a lot of methane in the sediment and where there is also abundant nitrate and preferably little oxygen. These are the current field locations: urban waterways, Amsterdam Canals, Twente Canal, Ooipolder, Brunsumer Heiden, Lake Grevelingen, we were there last weekend, Vercelli paddy fields, and I was fortunate to go twice to the Bothnian Sea this year to collect samples. In an ideal situation, the ecosystem should look like this. This is Brunsumer Heiden. Here is the groundwater level, the top. If you go down into the soil of Brunsumer Heiden, 50, 100, 200 centimeters, what you will see is from the bottom, there is from 160 centimeters methane production, reaching a peak around 120 centimeters. But when the methane comes in contact with nitrate-rich groundwater, which is very high in Brunsumer Heiden, you see that both methane and nitrate go down. This would be an ideal place to look for nitrate-dependent methane oxidizers. And such a soil you could take to the laboratory, do activity tests, try to enrich and sequence their genomes. Then you put this soil into a bioreactor, you feed it with methane, nitrate and some nutrients. And first we did the first enrichment we did from Twente Canal sediment in 2006. Later on the Ooipolder, Vercelli soils, and at the moment we are busy with Bothnian Sea sediments. And since we now have the diagnostic tools, what you would see is that it takes quite a while before you develop a good enrichment culture. 1.5 years is not an exception. It requires a lot of patience from the PhD candidates. And then what you will see is that you develop, in this case, anaerobic methanoxidizers that are archaea, old microorganisms, and we also have some bacteria that can do the trick. Now, what do these anaerobic methanoxidizers do? They convert methane via an intermediate product to CO2. 8 electrons are generated, and they use these 8 electrons to reduce 4-nitrate to nitrite, and therefore they always live in symbiosis with nitrite consumers. This is how they look like. Same as Anamox, very red microorganisms full of cytochrome Cs, and we could detect their diagnostic enzyme, methyl coenzyme M reductase. In the meanwhile, we have sequenced their genomes and have a reasonable picture what their metabolic secrets are. We have also elucidated their proteome, and indeed they do have methane activating enzymes. Indeed, they do have nitrate reducing enzymes. But as I said, what they do is they oxidize methane to CO2 and generate 8 electrons. And same as for Anamox, what we could do is harvest these electrons and make current, light, electricity. And that's what Helene Obouter, a current PhD candidate, is doing. She put some of the biomass in this microbial electrolysis cell, an anode, which she inoculates with this enrichment culture. And then she could see, yes, the cells are alive and kicking. They convert methane with nitrate. If you remove the nitrate, the cells will still oxidize the methane, but then donate the electrons to the anode of this system, and a current will develop. In previous times she was using carbon cloth as an anode. Nowadays she's got more expensive. She uses gold electrodes. And if you put these gold electrodes under the electron microscopy, then what you can see is nearly a pure culture of this Methanoperedens archaea of the anaerobic methanoxidizers. So it turns out also to be a very good system to enrich for one peculiar organism. And recently she published that, and you can see journalists are not all, it's not a bacterium but a microbe. Well, let's not be semantic about it. This bacterium can make electricity from methane, and also Arjan Dijkgraaf wrote a nice feature in Chemisch Weekblad. So we have discovered these new impossible methane oxidizers, nitrate, AOM. You need high patience because they grow very slowly. But didn't tell you high methane partial pressure does help, and you can make electricity at low temperatures. Now the last point, we have a ESC synergy together with Caroline Slomp from Utrecht University, and there we want to see if we can find iron-dependent methane oxidizers in coastal systems. Yeah. It's called MAREX, methane and ammonium removal in redox zones in coastal systems. In some of these coastal systems, there is a large burial of nitrogen oxides or metal oxides, and we think that the burial of these oxides can help to prevent methane escape, which is made deeper in the sediment, and these organisms are there. With the help of nitrate and metal oxides, they can prevent the methane from escaping. So we have chosen 3 field site locations, from Oxic in the Bothnian Sea to hypoxic Lake Grevelingen, an intermediate Stockholm Archipelago. Also oligotrophic, the Bothnian Sea, to very eutrophic in Grevelingen, so we have a gradient of different environmental conditions. And luckily we also have some preliminary data on the conditions in the field. So 4 weeks ago we were privileged to go with the, you can read it, all of us understand this Swedish, with the Coast Guard of Sweden. They have a very big ship, so we could sail the Bothnian Sea. They were very relaxed. I don't hope there are Russian submarines in the Bothnian Sea, because I'm not sure if they would have detected them. But they were very hospitable and good food. What we could measure there is what you see in the Bothnian Sea is that sulfate is very quickly depleted in the top layer of the sediment. There is a lot of methane being produced deeper in the sediment, and there is a lot of reactive iron deeper in the sediment. And so this would be an ideal zone to look for iron-dependent methane oxidizers. Well, that's what we did. We took this layer at 30 centimeters, put it in bottles, put a high pressure of methane in it. Then the control, no additions, then you see not much happens. If you feed only labeled methane, then the iron oxides still present in the sediment act as an electron acceptor. But when you give a lot of fresh metal oxides, iron oxides, you see a rapid on-conversion of the methane into CO2. When the iron is depleted, it stops. If you give a fresh new injection, it continues. And this coincides when we look at the genetic material, we see a lot of these anaerobic methanoxidizing archaea signatures in the sediment. Now, recently we started to look at Lake Grevelingen. Now maybe if you take a sediment core from Lake Grevelingen, you maybe can already see the many holes in this core. The sediment is really super saturated with methane. If you take a core, the methane is oozing out of the sediment. And if you slice the sediment, that's what Anna is doing here, slice the sediment, then you see there is an enormous methane production in this sediment. We can also find genetic material of methane-producing archaea, and not too much of the methane-consuming archaea, which also means that it is not a very good methane filter. And unfortunately, we can confirm that Here is a methane flux meter, which you put on the water. Luckily, Lake Grevelingen is still a CO2 sink. So you see in red, the CO2 is going down if you measure over, 10 minutes. But every season we have measured a methane efflux from Lake Grevelingen. So the methane in the sediment comes in the water column and goes up in the air. Now the last, I'm nearly there. Last ecosystem, Amsterdam Canals. There we are investigating 3 spots. There is a salt gradient which we thought would be influencing the methane emission in Amsterdam Canals, Magerbrug, Mariniersbrug, Zoutkeesgracht. Unfortunately, all of these points, you'll see there is a high flux of methane, and even this intermediate point has the highest flux of methane out of the sediment. Not so good. Well, to end on a good note, then, in the sediments there is a methane biofilter. If we take a sediment core from the Amsterdam Canal, slice it, and then add different electron acceptors, we can see there is some activity with nitrate, some activity with iron, not much with sulfate. They like graphene oxide very much. Graphene oxide is we use that as a homologue for humic acids, but no additions, not much happens. And you can also see the deeper you go into the sediment, the more methane is being oxidized by these anaerobic microbes. And indeed, high abundance of methane-producing archaea, somewhat high abundance of archaea that can eat the methane. Yes, nitrate-dependent methanoxidizers exist and they can help us remove methane from ecosystems. We have enrichment, they can make electricity. A lot of iron-dependent methanoxidizers are present in marine and freshwater systems. And one of our next challenges together with PAC and TTW is to apply this in a wastewater treatment system that would prevent the buying of the methanol. These organisms can use methane to remove nitrate, so we don't have to buy expensive methanol. But still some challenges there. In the past 20 years, I've spoken to the press numerous times. Not all of them have a biological background. And Fox is not my favorite news broadcaster, but Jenna Brenner had it right. New anaerobes could save the world. And with that, I would like to thank you for your attention.
Are there any questions for Mike?
Yes, please. Mag ook in het Nederlands. Dan vertaal ik het.
You mentioned that there is a way, microbial way to prevent the methane from the permafrost layers to come up. Or did I understand that correctly?
I wish there were a... Now, if the permafrost warms, the methane will be released. There is a foundation, it's called the Permafrost Foundation. They have called me and they said, Mike, how many methane oxidizing bacteria should we spray on Alaska permafrost to prevent? And could you cultivate them? Well, when we did some back-of-the-envelope calculations, I don't think you can cultivate enough bacteria to spray in this field. So the best way is to keep it cool and let the methane stay there.
So in principle it's possible, but it's not practical.
Not practical.
Yeah.
Not, yeah. I would say also stimulate, if the hope is that if the warming is slow, the organisms adapt and also become faster in eating the methane. Yes, please.
There's also a problem with this methane knollen in the deep sea. Yeah. Did you ever investigate those systems? Or?
The question is, I translate it as methane hydrates.
Yeah, methane hydrates.
Yeah. Also, the deep sea is about 2 degrees, but also the ocean is warming up, and the risk is that also these methane hydrates will collapse, and then you get an enormous methane bubble. No, well, to investigate those systems, you need really big ships, And even the Netherlands Sea Institute doesn't have such a big ship. So the Germans, they have the Polarstern, or the Americans, they have big ships. They investigate these deep sea sediments and the methane hydrates. Yeah. And there are organisms there that do eat methane hydrates. Yeah. But they do it very, very slowly. Yeah. Pete, yeah.
I would like your comments on new anaerobes could save the world.
Yeah.
Do you agree? Under what conditions is
Well,
this right?
Maybe not the whole world, but I hope they have shown you that that we have already been that it's already possible to make our wastewater treatments more sustainable, less emissions, more sustainable, so they at least contribute. Yeah. And with I said I should have brought the news release with me for in Wageningen they have discovered a new gut bacterium, and they're now really producing this gut bacterium for people who have a problem with their diet to sell it. So yes, small steps, but we as humans are there to save the world. The anaerobes can only help us. We should adapt.
Are your findings being accepted by the world of the wastewater treatment companies? And if not, what is the reason that they don't embrace it?
In the past 20 years, they have become much more progressive and less conservative. And also the EU, in this case, I think the EU helps. When the discharge standards become more and more strict, there is an incentive to do something. So and the nitrate emission standards have been going down from 50 to 10 milligrams nitrate per liter. So they needed innovative new wastewater treatment systems to remove the nitrogen compounds. And if there is legislation or a financial pressure, they will innovate. Yeah.
Maybe a similarity with the carbon dioxide.
Yeah.
People say that if you price carbon dioxide, then suddenly there's an incentive to
Yeah.
to a lower level.
Adjacent to one of the ERC programs, we have done an ERC proof of concept. There we have done a feasibility study on several processes, and also what the levy should be on methane emissions to make these things commercially viable. There are no levies on methane at the moment. I'm sure in 10 to 15 years they will be there. Yeah. If these levies come, then these processes become economically viable, but not at the moment. Yeah. Sander?
The Methanoperedens using the nitrate and attaching to the goal.
Yeah.
Are they sturdy microbes or really vulnerable to external input besides the oxygen?
Yeah. Sander's question is whether they are very sturdy. Maybe you can be more precise. You can squeeze them. You can mechanical pressure. That's no problem. One drop of oxygen and they are dead.
But if they take a long time to grow,
Yeah.
you, for instance, tap the glass and they already.
No. We can I think Helene is now operating the systems for more than 100 days. So that if you take care that there is enough methane and no oxygen, then it's quite stable.
Oh.
Yep.
So no light influences.
Oh, she keeps them in the dark. Yeah. Good. Yes, please.
I'm certainly not an expert, so maybe
Yeah.
a very stupid question.
There are no stupid questions, and I didn't explain it off.
I'm intrigued by the nitrogen chemistries.
Yeah.
Is there anything in your research that you can apply it in a new way of thinking of fertilizing crops? Maybe that I see all these conversions. Well, can you? It's also clear that let's say the ammonia fertilizers are really inefficient. Huh? So, and I also heard about microorganisms that they can maybe turn locally available substances into something that can fertilize, improve, let's say, the fertilization of crops. Is that related to this work, or is it completely different subject?
Both.
It's a nice answer.
No, I will explain. What I said, in the future, you can convert ammonium into dinitrogen gas. That is sustainable, but not the most sustainable thing. It's better to capture the ammonium and use it as fertilizer. So we have colleagues in Delft and in Groningen who are studying these processes. How can we capture the ammonium from wastewater and use it as fertilizer? then in Wageningen and John Innes Center in the UK, people are you have microorganisms that can fix nitrogen, so they fix dinitrogen gas from the air into ammonia. They live together with legumes, vlinderbloemigen, that is beans, peas. But what you really would like is corn or potato to be nitrogen fixing. And so people are trying ways either to manipulate bacteria to live in symbiosis with corn or potato, or even one step further is to transfer the genes of these bacteria to the corn or to the potato so that the crops themselves can fix nitrogen. Yeah, so this is ongoing work, and some of them are quite far. Maybe one step further would be that we get nitrogen fixing genes, and then we can cut out the meat. Yeah. Yeah. And another point is, and a long time ago I also worked on genetic engineering of amino acid production. What we and our husbandry lacks is lysine and methionine. These are two essential amino acids, so we try to produce that with bacteria. Yeah. Yeah. Yes, please.
So you mentioned several times that it's not easy to scale it.
Yeah.
So what's the main challenge in scaling up either the growth or the application?
Now, luckily with the Anamox, it took 2 or 3 years to scale it up. And it was not the biology that was the limiting factor, it was the engineering of the system. Very trivial things that even in the Netherlands it can be -20 in winter and then a pipe froze. But we have solved it for Anamox, and I think for the other processes we will not make the mistakes again. Yeah. But if the biology works on 1 liter, it also works on 100,000 liters. Yeah. And maybe we should become an engineering faculty. Or at least have engineers that help us. Nowadays we go always to Delft. Yeah.
You at least answered one of their questions.
But and without, we didn't talk before, so.
Yeah.
More questions? Now is the moment.
Okay.
And otherwise it's my last day of the week, so I like a beer.
Thank you so much.
You're
That was
welcome.
great. Thanks everyone for coming. We have beer and wine outside for half an hour. Feel free to speed it up. Feel free to hang out a little bit after, but just please will be gone at 9:30. So Mike, I want to give you this for tonight. Thank you so much.
Dankjewel.
And
Anaerobic fermentation by a eukaryote.